Hello, everyone, and welcome to Advances in PSC Workflows to Build Better Models and Therapies. I'm Matt Thorne of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. We do encourage you to participate today by submitting your questions you may have during the presentation. To do so, please type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You can also submit any technical issues as well if you're having trouble seeing or hearing, and we'll contact you directly through that Ask a Question box. I'd like to welcome our speaker, Dr. Omar Farah, Manager, Field Application Scientist, and Regenerative Medicine. For the complete biography on our speaker, please click on the biography window on the left of your screen. Dr. Farah, you may now begin your presentation.
Thanks, Matt. Hello, everyone, and thank you for joining today's webinar entitled Advances in PSC Workflows to Build Better Models and Therapies. My name is Omar Farah, and I lead a team of regenerative medicine field application scientists at Thermo Fisher Scientific. And today I'm going to be going through an overview of the stem cell workflow, highlighting new advances in data to help better build PSC models and hopefully systems and therapies. So pluripotent stem cells, or PSCs, are remarkable cells that originate from early stage embryos and have two defining characteristics: the ability to self-renew indefinitely and the capability to differentiate into any cell type from the three different germ layers, whether that be endoderm, mesoderm, or ectoderm. And so what this basically means is that they can give rise to virtually any cell type in your body. And the true power of PSCs really lies in their regenerative potential, which has opened up a myriad of applications. In basic biology, PSCs help us understand early human development and cell differentiation processes, while in disease modeling applications, they allow us to create accurate models for human disease, enabling better understanding and treatment strategies as well. Now, for drug discovery applications, PSCs provide a reliable platform for screening new pharmaceuticals, but also for cell therapy applications, PSCs hold a big promise of generating healthy cells to replace damaged cells or tissues, offering potential cures for various different debilitating diseases as well. And as we dive a little bit deeper into the world of PSCs today, we'll explore these applications and their transformative impact on science and medicine. Now, over the past decade or so, Thermo Fisher has been working on developing solutions and reagents to support the entire PSC workflow, making sure that we consider both upstream and downstream aspects of this particular workflow to help ensure a continuous flow from one step to the next as well, whether that be from your somatic cell collection and culture to reprogramming, PSC culture and expansion, characterizations of these PSC cultures, and even genome editing and modulation of these PSCs before differentiating them into a specific cell type of interest. And so today we will focus on PSC culture, on the PSC culture portion of the workflow and how it affects downstream processes as well. But before I do that, just a brief introduction or history is, as many of us know, the stem cell field has made tremendous progress when it comes to how we culture and cultivate PSCs. We started off with feeder-dependent cultures that were supplemented with either FBS or knockout serum replacement, and slowly, progressively moved towards more defined feeder-free media systems that can, be paired up with different substrates like vitronectin, or laminin or different basal membrane extracts to help culture these cells in maybe a more favorable condition for their growth and subsequent differentiation. But more recently, our team at Thermo Fisher developed our StemFlex medium, which is a robust PSC media that was specifically designed for supporting challenging workflows with PSCs, workflows like gene editing, single cell sorting, and clonal expansion as well. And so what our team did was we used an iterative design of experiment approach coupled with follow-up titrations to really fully optimize the StemFlex media system. With me showing you one such experiment that's outlined in this slide here. But we tested over 100 different formulations to completely optimize the StemFlex media system with this DOV approach. And what this basically allowed us to do is to identify one at a time, these deleterious components, the critical components, as well as what we call neutral components in the formulation, and essentially allowing us to optimize the formulation of the media to something that would be super supportive for the cells without including anything that's not really essential. But I should mention that equally as important, for the performance and really of these cultures and the improvement of these cultures is consistency and manufacturability as well. So identifying how these individual components also interact with each other was a design element that was considered here. As well as identifying the critical concentrations at which these components should be added. So overall, in doing so, what we were able to do is reach a final formulation that was very supportive of PSC, especially during challenging workflows. Now, as many of you are well aware, PSC maintenance with traditional media required daily feeding. And this was mainly due to the thermal instability of FGF2 within the formulation of most PSC medias, where FGF2 concentrations would dip drastically within the first 24 hours. As you can see over here, resulting in really a need for these daily feeds to resupplement the media and the cells with FGF2. Now, with more modern media like essentially Flex or StemFlex, we were able to stabilize the FGF2 activity at 37 degrees for up to 72 hours, avoiding those really drastic drops in concentrations that can play a big role in how these cells differentiate or maintain their pluripotency. And so by doing so, in addition to having a more consistent FGF concentration during the culture period, what this allows us to do is employ a more flexible feeding schedule, where researchers can skip up to 3 different days a week of feeding, 2 of which actually can be consecutive skip days if we double feed on the day prior. But basically what this helps do is reduce handling times, reduce contamination risk, but also reduce the cost associated with the media that's used to culture here. And I can also say that, or I should say, I can confirm that we've done extensive testing confirming that this flexible feeding schedule, including weekend free feeding, does not impact either the pluripotency or the karyotype of our StemFlex PSC cultures. So as you can see here, even with these flexible feeding schedules, we observe high maintenance of pluripotency, both earlier passages, as you can see here, passage 6, but we've also done this work all the way up to passage 50 in StemFlex cultures. Now, I briefly mentioned that StemFlex was specifically designed to help address and support challenging applications within the stem cell workflow. And one such application is the clonal expansion of cells, either before or after gene editing. Now, you can see here that we've performed an experiment where we've sorted and seeded single cells in a single well of a 96-well plate in multiple different conditions, in 3 different conditions, I should say. And what we were aiming to do is to examine and analyze which of those 3 conditions would be more favorable for the ability of these single cells to generate a colony, i.e., the clonal efficiency of each condition. So you can see in the first condition, where cells were grown in traditional PSC media matrices like Essential 8 or Geltrex, that we see the clonal efficiency, meaning that the ability of that one cell to derive an entire colony sits at around 5%. In the second condition, the cells were maintained in Essential 8 media, but were also supplemented with the Revital Cell supplement, which is our version of a ROCK inhibitor. And we're also grown onto a laminin-based 521 matrix that is more supportive for the attachment of PSCs. And in those conditions, we see that the clonal efficiency with these particular conditions is closer to around 15%. But in the third condition, where we've sorted single cells and plated them back down in StemFlex media with the Revitacell supplement and the laminin-based matrix, we observe our clonal efficiencies in these conditions to be closer to 40 to 45% as well, really allowing you that flexibility in terms of how many different clones you can easily generate after a sorting experiment or gene editing experiment. And in fact, we've even done some direct comparisons with StemFlex media with other media systems where we consistently see a benefit to the utilization and culture of our cells in StemFlex media specifically over other medias. Again, specifically when it relates to very challenging workflows here as well, where in single cell passaging workflows, we see up to a 5-fold improvement in clonal expansion after single cell passaging or sorting. And this is even in the absence of a ROCK inhibitor. But also, we see that StemFlex supports gene editing workflows really well, as you can see here on the right, where we see up to a 2-fold faster recovery of these cells following a gene editing experiment as judged by cell doubling times. Additionally, we see improved survival and decreased spontaneous differentiation of our cultures, again, compared to other media systems that are currently used for PSC monolayer culture. Now, with the benefits that are conferred by StemFlex media, we can now streamline the generation of genomically edited lines and kind of the creation of different disease models. And that's exactly what we did at Thermo Fisher, where we streamlined the process where we can deliver our gene editing tools into our StemFlex cultured PSCs via electroporation with our Neon NXT electroporation system. After which we allow the cells to recover in StemFlex medium and potentially start our clonal expansion process before we screen and select a particular clone that we want to be working with. And this is actually, in fact, the process that we use to generate several different SNP disease model cell lines that you can see over here, where some of those disease models are related to CNS disease, these top two that are related to epilepsy and schizophrenia. But other cell lines where we've introduced SNP mutations into the TMT2 gene can be used to model dilated cardiomyopathy. We've also generated different lines that have SNP edits in either the SNCA gene or the LRRK2 gene that are commonly associated with Parkinson's disease as well. So overall, and depending on the location of the edit intended, we do observe anywhere between 20 to 80% indel formation or 5 to 40% SNP conversions with our particular protocols. Now, since optimizing our gene editing and disease modeling generation process with the StemFlex cultures, we've also introduced some other supplements that can be added to further aid in this process. And culture supplement is one such supplement that can be added. This was a supplement that was originally developed out of the Stem Cell Translations Laboratory at the NIH, where researchers over there screened over 15,000, compounds to identify a combination of small molecules that we commonly refer to as sept right now, to work together and provide the added support for a variety of different cell types, iPSCs and PSCs included. And so in various different peer-reviewed publications, NCATs have shown that the sept molecules or the sept cocktails can protect a number of different iPSC cell lines and ES cell lines, where it has been shown to block different stress mechanisms, including detrimental cell contractions, membrane blubbing, abnormal nuclear morphologies as well. But not only that, but sept also minimizes oxidative stress and avoids the activation of the integrated cell response system, really promoting cell health and attachment as well. Now, to this end, we wanted to see how the culture supplement can be used to improve cell attachment in these clonal expansion workflows that I described earlier. So we repeated a very similar experiment where we seeded 96 well plates, where we seeded single cells in 96 well plates. In this case, in very lean conditions with essentially media, where the only two difference between the two experimental paradigms here was whether the supplement was added was a traditional ROCK inhibitor in the form of the Y27632 compound or the culture sept supplement. And as you can see, the culture supplemented wells showed a significant increase in clonal efficiency compared to the traditional ROCK inhibitor, even in those very, very lean essential 8 conditions as well. Now, we've discussed StemFlex medium and its benefit to 2D or monolayer culture of PSCs, but I wanted to shift gears slightly here and talk a little bit about suspension culture and or 3D culture of pluripotent stem cells. So in a very similar design of experiment or DOV approach to the one that we used to develop the StemFlex medium, our R&D team developed another media called StemScale PSC suspension medium, which is a scalable, easy-to-use media that supports the robust expansion of PSCs, delivering anywhere between 5 to 10-fold expansion per passage. So this particular system uses a spheroid self-assembly method to promote PSC aggregation and growth, as you can see in the image in the bottom over here. And it actually doesn't require the use of any microcarriers or ECM substrates. And beyond the expansion capabilities themselves, the workflows that are associated with this media, whether that be media changes or passaging, are straightforward, simple, straightforward, and for the most part, amendable to either multiple different vessel formats and scales or different automation platforms as well. And this is true for across multiple different cell lines that we've tested. Now, beyond the actual media itself, there's 4 critical components needed to successfully culture PSCs in suspension. The first is a form of agitation, which we usually use a CO2-resistant shaker to provide that agitation needed to keep these cells in suspension and avoid cell clumping. The second is a dissociation reagent. In this case, we recommend the use of StemPro Accutase in order to dissociate either 2D cultures to initiate those 3D stem scale cultures or during passaging of our stem scale cultures. And we use the StemPro Accutase here to generate a single cell suspension in order to drive the progression or the passaging cultivation of the culture forward. Another component that you need here is actually a ROCK inhibitor. And in this case, we've tested several different ROCK inhibitors, but the addition and supplementation of the Y27632 compound specifically during the initiation of these 3D suspension cultures allows for the nucleation of these cells into spheroids. So this addition is very, very important. And then finally, of course, we need some sort of culture vessel format. Usually non-TC treated plates or flasks are used in small scale, but we also have recommendations on larger scales, including bioreactors, different types of bioreactors. And so initially, the stem scale suspension cultures are initiated by dissociating your 2D cultures or your adherent cultures with the StemPro Accutase into a single cell suspension, and then counting and reseeding at 100,000 cells per ml in your stem scale media with this ROCK inhibitor, with the Y27632 compound, either in plates or flasks, depending on the scale you want to be working at. And then those cultures can be placed on the CO2 resistant shaker in the incubator overnight. Now, upon the initiation of the culture, single cells will nucleate into these free-floating spheroids, like I mentioned, where the spheroids will be around 100 to 150 microns in diameter on day 1 over here. And as these spheroids continue to grow over the next 4 days, we recommend utilizing a 50% medium replacement strategy every other day in order to resupplement the media with the nutrients that are needed so that the cells are well supported, but also to remove any metabolic waste that is accumulating and maintain a constant volume of your culture that's going to be important to maintain a homogeneous size of your spheroids as well. Now, I did mention that when the spheroids continue to grow for about 3 to 4 days, they will reach a size or a diameter of around 400 microns at around day 3 to day 4. And that's a time where we generally recommend passaging these cultures via enzymatic digestion with this temporoacutase again. Here, during this process of passaging, there's no cell strainers needed, which increases your chances of, or decreases your chances of contamination, decreases chances of cell loss, and is also, the fact that we are not using cell strainers here makes it a little bit easier to and more amendable for this process to be included in automation platforms as well. Now, generally, the size of the spheroid, and I did briefly touch upon this, is an important factor during suspension culture, as it can really dictate, how the nutrients or gases are able to penetrate all the way to the middle of that spheroid, and really prevent the formation of either a necrotic core or losing or loss of pluripotency. So, generally speaking, spheroids grown in the stem scale media tend to grow at a very homogeneous rate, as you can see in these representative images from 4 different cell lines, 2 IPS and 2 ES cell lines. And typically around day 4 is when they reach that 400 micron mark, after which we recommend passaging those cultures. Now, one important design element of the stem scale media was really the ability to expand PSC cell lines, both ES and iPSC cells, especially when we consider how these suspension cultures can be really, really utilized in order to generate a large number of these cells that would be used for clinical or therapeutic purposes. So in this case, I wanted to show that with the different cell lines here that I've shown previously as well, the same 4 lines, we can see anywhere between 6 to 11-fold expansion per passage. So what this basically means is that our cumulative fold change over multiple passages increases quite dramatically. And you can see this in this example where only after 3 passages with those same 4 lines, we can see anywhere between 18 to 28 fold expansion in just about 15 days or so. Now, another really important design element of the media system was scalability. And again, this is especially important for these translational and clinical applications where you need to scale up your operations from smaller formats like 6-well plates, for example, to larger flasks or even bioreactors. And scaling across these formats it really is an important consideration because it can be cost effective and efficient. You need to be cost effective and efficient when transitioning from one vessel to the next. And here we show that the maintenance of these expansion capabilities of the media and the media system is really amendable across multiple different culture vessel formats. Whether you're culturing your cells in smaller volume shaker flasks and scaling up to 1-liter shaker flasks, or if you're starting off in smaller 6-well plates and scaling up into medium-sized bioreactors as well, we see a consistent performance of the media in terms of expansion capabilities that it provides to the cells in culture. Now, not only that, but you can also see a consistency in terms of the performance of the media over time. So on the right-hand side, we're showing you a longer-term study where these stem scale PSC cultures were maintained for 25 consecutive passages, where at each passage we were examining the expansion potential from the passage previous. And you can see here that the linear line across the 25 passages suggests that we were continuously getting that at least 10x or above expansion capabilities per passage that we've previously seen. Now, one common concern for a lot of researchers when culturing PSCs in general let alone in a newer suspension-based system, is whether that system can maintain pluripotency of their cells and assure a normal karyotype. And so we did a lot of extensive work to show that we can maintain above 90 to 95% pluripotency of our cultures, even after 30 consecutive passages in suspension, as as assessed here by flow cytometric analysis of OCT4 and NANOG. But this pluripotency status was also further confirmed by our pluritest array, where we are looking at global gene expression of our stem scale cultures and comparing them to a database of over 250 plus validated iPS cell lines, looking at the global transcriptomic signatures of PSCs and seeing whether they match what we known to be a nature or a transcriptomic signature of PSC culture. And thankfully, again, even after 30 consecutive passages in suspension here, we do see our stem scale cultures cluster closely to our validated iPSC cell lines in our database. Now, additionally, no karyotypic abnormalities were observed, again, even after 30 consecutive passages in the stem scale medium, whether we were judging it by our karyostat SNP array to look for chromosomal abnormalities, or or judged by our Oncomine NGS assays, looking for even specific point mutations in proto-oncogenes known to be commonly mutated in iPSCs. So overall, kind of the take-home messages here is that we can really successfully generate a large number of PSCs with the stem scale suspension medium for downstream applications as well. And in my mind, at least, this is where the fun really begins, where we start to differentiate these 3D spheroids that we've generated into lineage-specific cell types. And to this end, our R&D team has generated several protocols and guidance and how we can take those iPSC-based and the suspension-based cultures, those iPSCs grown in a stem scale medium, and differentiate them into a bunch of different cell types and organoids without the need for an embryoid body formation step as well. So in the next several slides, I'll be going over a couple of examples or several examples where we've successfully differentiated our stem scale cultures into several different cell types and organoid types. The first example here is where our stem scale PSC spheroids that you can see on the bottom over here are differentiated into floor plate progenitor spheroids and then eventually into midbrain organoids using our PSC dopaminergic neuron differentiation kit. And so in just 11 days in a 3D optimized protocol, we've arrived at a phenotype and a cell number that is very comparable to day 21 of the traditional 2D protocol, where on day 11, as you can see over here, those day 11 progenitor spheroids, looking at their phenotype, they stain over 90% positive for markers like FOXA2 and OTX2, excuse me, confirming their floor plate progenitor state. But not only that, so after day 11, where we specified progenitors, initially we dissociated these progenitor cells down and plated them back down into 2D in order to mature them into TH expressing dopaminergic neurons. And we were able to actually specify TH expressing dopaminergic neurons in as early as day 21 of our protocol. And again, this is in stark comparison to the traditional full 2D protocol, where you can only achieve this at day 38 as well. And so I mentioned that for the maturation portion, we started when optimizing our protocols, we started by plating those cells back down into 2D. But we also wanted to test whether the maturation of these, of these cells can be done in suspension or in 3D as well, from start to finish. Again, really eliminating that need to culture these cells back down into 2D for that maturation step. And here we've tested multiple different conditions for that maturation portion, where rotating cultures and parameters, as well as different ECM supplementations and encapsulations were evaluated. So I'm showing four different conditions here, but what I wanted to really draw your attention to is the last column, where we've noticed that the maturation of these midbrain organoids in a static 96-well U bottom plate format, where we've added around 2% gel tracks to the media, provides the best results for both scale-up and the derivation of TH positive, MAP2 positive expressing midbrain organoids. Now, one very interesting observation that we made during the differentiation of these midbrain organoids in 3D is neuromelanin deposition, where we saw a deposition of this brown substance, if you will, in those mature midbrain organoids. So, for those of you who may be not very too familiar, but neuromelanin is a pigment that is found in the substantia nigra, where these dopaminergic neurons usually develop in vivo in our brain. And so this deposition is rarely seen in 2D culture, but we started to see it a little bit more easily in the different 3D maturation conditions that we've tested, as you can see here, where especially as you can see on the right, that condition that I called out in the last slide, that 96-well U-bottom plate where the cells are being cultured under static conditions, provides the best the best deposition phenomenon that we've seen. And so what this data really suggests is that our 3D differentiation models can potentially provide a more physiologically relevant models in certain instances that may be better able to predict or mimic human biology as well. Now, interestingly as well, we wanted to compare the functional activity of these midbrain organoids. We placed them on multi-electrode assays to measure their electrophysiology and actually observed very strong functional activity when they're plated on these multi-electrode assays, especially compared to 2D differentiation protocols, as you can see in these two top raster plots. Now, whether we're looking at mean firing rates, synchronicity of our cultures, or even just the number of active electrodes, what we've seen is a pattern where the 3D differentiated cultures always tend to outperform the 2D differentiated cultures that you see in the gray lines as well for all three plots. So again, this is just another example of how utilizing such a system can help you to potentially develop a more reliable and physiologically relevant model to help address or recapitulate human biology. Now, ectodermal or neural lineages are not the only cell type that we can specify or differentiate into with our stem scale PSC cultures. Here's an example where we worked with the Lee Lab at Harvard to optimize a protocol for the differentiation of these PSC spheroids into beating cardiac organoids, or cardioids as we like to call them, in just about 2 weeks, where by the end of this 2 weeks, you'll have these beautiful beating cardioids in culture. Now, this protocol utilizes small molecules to stimulate Wnt activity initially, driving the transition into mesoderm before eventually blocking Wnt activity yet again in order to further induce a cardiac-specific phenotype. And you can see here that we can achieve really high purity of TNNT2 positive cells with this particular protocol of over, in this case, over 90%. But I should mention that the concentrations of these small molecules are very important and may need to be optimized for the particular cell line that's being used as well. But with that being said, after the initial first 2 weeks of differentiation of these cardioids, we can continue to culture them in suspension, allowing them to further develop and mature. And in fact, when doing some preliminary characterization of these more mature cardioids with high content imaging platforms, we actually do observe the specification of alpha-actinin and cardiac troponin, cardiomyocytes in these organoids that tend to really nicely organize or self-organize somewhere in the middle. But we also can detect fermented positive fibroblasts, as well as CD31 endothelial cells that are shown to be present in close proximity and sometimes even surrounding those cardiomyocytes as well. Now, finally, similar to what we did with the midbrain organoids, we wanted to see whether we can assess the functional characteristics of these cardioids. So we took these cardioids that were differentiated in a small-scale bioreactors and plated them onto multi-electrode assays to measure again their electrical output. And here we can see, the visible beating of the cardioids displaying rhythmic voltage pulses. And our analysis shows an extremely stable beat rate of 0.3 seconds, for example, in some of those cardiac organoid models. But aside from all of that, even on a high-resolution multi-electrode assay platform, we're actually able to detect repolarization T waves following depolarization, as you can see in the top right corner over here, which again, altogether, we believe this provides a really valuable step for cardiac disease modeling, where now we have different tools and models to study things like arrhythmias or even dynamics of depolarization and repolarization as well. Now, in the last example, I wanted to highlight something slightly a little bit different, where in an effort to support iPSC-derived cell therapies, excuse me, Thermo Fisher has optimized a scalable protocol for the generation of induced natural killer cells from PSCs that were grown in our CTS version of the stem scale PSC suspension medium. So the CTS stem scale medium is formulated and designed to be of the version of the original stem scale medium, but that is specifically geared towards use in clinical or translational applications. And as part of the cell therapy systems grade of or brand of products and reagents and instruments, CTS stem scale provides similar performance to the research use only product, but is also formulated specifically to facilitate this easy transition into those clinical manufacturing settings where considerations have been taken around reducing risk when it comes to raw materials or components, increased safety testing, dedicated quality support, as well as GMP manufacturing, of course. And so in this particular protocol, we outline the differentiation of PSCs into NK cells using our CTS stem scale media in just about 5 weeks or so, where we slowly transition those CTS stem scale spheroids that you see here into a hemopoietic progenitor state in about 9 days by culturing these cells in different basal medias and different pepper-tech cytokines that are going to help with this initial induction into a hemopoietic state. And then following that, we will further specify these hemopoietic progenitor cells into a lymphoid and eventually a natural killer cell phenotype. Again, while culturing them in our CTS NK expander media, supplemented by MEM alpha and human AV serum, along with specific cytokines and growth factors that we know are going to help push the cells to that particular INK state. Now, throughout the IK differentiation protocol, specific changes can be observed, morphologically during this process. But there's also specific changes to the phenotypic profile of these cells that we can follow over the entirety of the differentiation protocol. So, for example, on day 9, we see the emergence of the definitive hemopoietic progenitors as indicated by the presence and expression of markers like CD34 and CD90. At day 21, the emergence of INKs is signified by the expression of markers like CD56 and CD16. And eventually, towards the end of the protocol, we're specifying cells that are CD56 positive, CD3 negative, and CD16 positive. So these are specifically natural killer cells, not T cells, that seem to have the capacity, the killing capacity or cytotoxic capacity, I should say. Now, after the derivation of these iNK cells, using the protocol previously shown, these iNKs can be further expanded and enriched in our CTS NK expander media that's supplemented with IL-15 for about 2 weeks. And so this enrichment period allows for us to increase the purity of our cultures, where we can increase the purity of the CD56 CD3 negative population, as well as increasing at the same time the incidence of the double positive CD56, CD16. These are going to be your cytotoxically active natural killer cells. And so overall, the main message here is that the iPSC-derived iNKs can selectively proliferate and be enriched in this particular media, where by the end of this enrichment or expansion protocol, we can see up to a tenfold improvement in terms of, or tenfold improvement to the ratio of iNKs to PSC specified. So for every one PSC cell that you start with, by the end of this expansion period, you can get up to, depending on the cell line you're using, up to 10 iNKs. Now, to gain a better understanding of the functional cytotoxic capabilities of these cells, we devised a co-culture killing assay using our OncoPro tumoroid culture media kit. Now, for those of you who are not aware, the Gibco OncoPro tumoroid kit is an easy-to-use culture media system that's been optimized for culturing and expanding patient-derived lines in a 3D setting, while maintaining the same mutational profile from the original tumor sample or patient sample. And in this particular experiment, CTS-derived iNKs that we previously just discussed were co-cultured with human colon GFP expressing tumoroid lines that are grown in our OncoPro tumoroid culture media. And so the goal here was to assay how much what is the cytotoxic activity that these effector cells, being the iNKs, can place on these target cells or our tumoroid GFP positive cells. And so you can see that the GFP signal from the colorectal tumoroids that's visualized in green over here significantly drops as soon as we add in our iNK cells to the culture system. And this is accompanied by an increase in caspase-based signaling, indicating cell death as well. That's visualized in red in the images on the left too. And while I should note here that the tumoroid targeting was best at a 10-to-1 effector to target ratio, really indicating that these iNKs can successfully target patient-derived tumor cells and kill them in a dose-dependent manner as well. And so to conclude, I would like to again highlight the tremendous progress that the stem cell field has made in the past several years to not only improve our understanding of human biology and disease and how we can potentially model that in vitro, but to also have tools that were able to push these workflows forward, especially when considering transitioning to clinical or therapeutic endpoints as well. And so I wanted to outline here an example of Thermo Fisher's iPSC-derived cell therapy-based workflow, where we've utilized our broad offerings of CTS products that are in regulatory and quality compliant offerings, whether that be from reagents to instruments, to really support the use of these PSCs in clinical and therapeutic purposes. We talked a little bit about CTS stem scale being one such media that you can use to transition from basic research into clinical applications for suspension culture as well. But this is also true throughout the entirety of the stem cell workflow, whether that be from somatic cell isolation and reprogramming to cell engineering and expansion before eventually differentiation and downstream steps. All of these particular workflow steps need really a stringent consideration on what materials, reagents are being used that are amendable for those clinical or therapeutic applications. And then finally, just to kind of end, I wanted to invite you to check out the plethora of resources that we've developed at Thermo Fisher to help highlight certain products or protocols or reagents that can assist you in your stem cell workflow. Not to mention that we have a ton of resources and support in terms of, in the form of free e-learning modules, how-to videos, handbooks, protocol books, as well as, field application scientists and R&D scientists on our teams as well. And with that, I will be happy to take any and all of your questions as well. Thank you for your attention.
All right, thank you very much. Thank you, Dr. Farah, for your informative presentation. We're now going to start the live Q&A. If you have not sent in any of your questions yet, I recommend going ahead and doing that now. We'll try to get through as many as we can before our webinar wraps up. So let's go with our first question here is, how did you optimize the payload delivery during the gene editing portion with the StemFlex medium?
Thanks, Matt. Yeah, we tested multiple different delivery methods, including lipid transfection with different lipofectamine reagents, as well as electroporation. And we've seen that electroporation usually gives the best results in terms of delivery into PSCs or generally hard to specify cells. Specifically with our Neon Next electroporator, we have the ability to optimize our electroporation parameters. With the Neon Next provides the customizable protocols where your electroporation can just happen quickly in your tip, and that seems to be the best way to do it. And we do have some recommendations on voltages, voltage width as well that should help with the delivery of different cargoes too.
Thank you. Let's see here. Is it recommended that the media for the stem scale cultures be changed every day, every other day? And can you do more than 50% medium replacement?
Good question. It's sort of along the same lines as StemFlex. So you can skip feeding with the stem scale cultures. So you can feed every other day. And in terms of the media replacement, 50% is the recommendation. You can definitely do more than a 50% medium replacement. What we've seen is that it doesn't necessarily have the largest impact on the health of the cultures past that. That being said, there are certain applications where you have to do, complete media replacements or something like that. If this is something that you're working towards, you can definitely do it. You just have to be careful that doesn't affect your cell yield. And specifically when we're talking about larger scale formats, with larger scale bioreactors, perfusion-based protocols are probably the best way to go.
All right. Let's go with how are the stem scale suspension cultures, PSC spheroids, passaged during the routine culture? And can the spheroid cultures be passaged before they reach 400 in diameter?
Yeah, in terms of the passaging with the stem scale cultures, we recommend the use of the StemPro Accutase again to take those spheroids back down to a single cell suspension or solution, after which you can just count and reseed the exact same way you would as when you initially specified or initiated those cultures. Now, the cultures can definitely be passaged before 400 microns in size, with the caveat that if they're not given that extra time to expand, the the cell yield might be slightly lower because you're harvesting, a day or a couple of days prior, but it can definitely be done.
All right. At what resolution does the cryostat characterization provide?
Yeah, so with the cryostat, this is a SNP array-based system. We analyze 2 million cells from a frozen cell pellet that is sent to our custom services group. But typically, in terms of the resolution, we can see up to or chromosomal gains and losses as small as 1 megabase pair. We can also detect the loss of heterozygosity, as well as have a mosaicism detection limit of about 30%, where we can tell different mosaic populations or subpopulations in our culture if they're present at 30% or more.
All right. We do have time for a few more questions. If you haven't sent one in yet, go ahead and do that. Just a reminder, anything we don't get to, we will follow up with after the event. So let's go with how do you manage working with a very large amount of cells during suspension culture and passaging?
Another good question. And just in terms of the number of cells, it can be a little bit daunting, especially when you're working with very large volumes. Um, uh, Typically, what we see at around the 3-liter scale is when we start to get about 5 billion passages, 5 billion cells per passage. And that can be a little bit hard to work with manually. So we have been developing some more closed system protocols in order to help with with that part of the workflow. Specifically, we've been using our CTS Rotea counterflow centrifugation system, which is an instrument that uses counterflow centrifugation to separate and isolate cells, but can also be used for different media changes. And in our case, in a very closed system, we're having our cells funneled into this instrument, and then the media is replaced with our Sempro acutase so that the spheroids can be dissociated quickly, and then brought back into, again, a single cell suspension for subsequent either passage or downstream applications as well. And I should mention here that we have done some comparative studies showing that viability, pluripotency, as well as fold expansion, that these metrics do not change whether we're dissociating or working on a smaller scale, or doing so with larger volumes or cell numbers with the Rotea system as well.
All right. Let's go with can I use the cell culture for HCMV and EBV cultivation to treat with some compounds?
I'm not sure what that is referring to specifically, to be completely honest.
Okay.
I'm happy to take this one offline and reach back out to that particular person. So
Okay. Yep. We'll pass that along. Can I use Revitacell instead of Y27632 to start the stem scale suspension cultures?
yes, I briefly alluded to this. We did test multiple different ROCK inhibitors and sort of their ability to initiate the stem cell spheroid culture. We call that particular process nucleation, where those single cells sort of aggregate and start to form a spheroid. And Revitacell was one of the ROCK inhibitors that was tested as well at the time. So I would say that even though Revitacell works great for adherent monolayer cultures and can be continued to use there, we found that the nucleation of those single cells into the spheroid is usually better with the Y27 compound. That also being said, I did mention the culture supplement, which also includes a ROCK inhibitor. We have tested the culture supplement in the stem scale spheroid nucleation process, and it does actually support that step pretty well. The only caveat there is we're still waiting for to complete long-term characterization data, similar to what we did with the Y27632, just to make sure that our cultures are karyotypically normal and don't harbor any chromosomal abnormalities or point mutations as well, with the SEP supplement too. So that's something that's yet to be done.
All right. So I want to thank you again, Dr. Farah, for your time and your important research. I'd also like to thank LabRoots and our sponsor, Thermo Fisher, for underwriting the educational webcast today. Before we go, I do want to thank the audience for joining us and for your questions. Any questions we didn't have time for today, and those that are submitted once it's on demand, they'll be addressed by the speaker via the contact information that you provided when you registered. If you'd like to request additional information, please be sure to fill out the survey. It's in the bottom left of your screen. It'll also be on screen after we end the webinar here, and someone will reach out to you. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available. And we do encourage you to share that email with your colleagues who might have missed today's live event. Until next time, thank you very much and goodbye.